Hi, everyone. Thanks for being here. I am Satrajit. I am a data scientist with the Institute for Data Science in Oncology at the MD Anderson Cancer Center. So the talk that I'm giving today is related to AI-driven workflow automation for neuro-oncology. This actually started as my final PhD project, and then it was in collaboration with between WashU and MD Anderson. And then I am in MD Anderson now, and as a part of the tumor measurement initiative, I am continuing to extend this in more directions. Today's talk will focus on the initial version that I developed at WashU, which focuses on segmentation of gliomas. So the motivation for this work was essentially the difficulties that researchers face when curating large-scale neuro-oncology data sets. Now, these data sets are very high dimensional because they are all imaging data sets and they are of high volume size. And these data is also very heterogeneous because they are acquired using varying acquisition protocols. So luckily for us, AI can actually distill this data into meaningful quantitative information. For example, if I talk in the context of neuro-oncology, we can go from MRI sequences, which are obtained using varying acquisition protocols, to a tumor segmentation mask, let's say, which can be used for treatment planning. But even though this path looks very straightforward, there are multiple intermediate steps that are essential for the success of these AI models. And these steps are curation. We need to curate the data in a way so that we get scans of specific contrast properties. We need to pre-process the data in a standardized way. And we also need to accurately segment the tumor. Now, there has been some efforts to automate these steps, but most of these efforts are fragmented, and the curation part is largely overlooked. So even though we often need like weeks or months to curate a data set before we can actually feed it into the AI model, but there has not been too many work to automate this process. And the reason is that each of these steps has their own set of challenges. For example, in the curation part, let's say if I talk about MRI data, then the scans of same MRI sequence can have different series descriptions because they are acquired using different acquisition protocols or they come from different vendors. So when I was looking at post-contrast T1 scan for my project, and this is the public data set I was looking at, these are all the different series descriptions that I came across. And you can imagine that it is very difficult to manually select the T1 contrast scan from each session with these varying series descriptions, and also it is very difficult to come up with some kind of heuristic to do this automatically. The second challenge is that sometimes we see that scans which are of different sequence can have same series description. For example, this sometimes happens for pre and post contrast T1. And the third challenge is that sometimes there are multiple scans of the same sequence and same series description, but some of them are poor quality and some of them are high quality. So when someone is curating the data, they actually need to go inside each scan and determine which scan needs to be used for the downstream task. For the pre-processing part, the manual pre-processing is very time intensive and tedious, and it can have some reproducibility issues as well. For the tumor segmentation, manually delineating tumors is very time-taking because when an expert annotator annotates the tumor, they have to often look at multiple different sequences to capture the complementary information that these sequences provide about the tumor. And they have to go slice by slice and annotate each slice. And this can be very time-consuming for big data sets. So for example, here I'm showing a high-grade glioma on a post-contrast T1 weighted scan. And this is the tumor segmentation mask that we want. And as you can see that this tumor is very heterogeneous, it's infiltrating. And there are multiple tumor sub-tissues that we are interested in. And if the annotator will need quite a bit of time to draw this, even on a single slice. And then they need to do this on all of the slices in a volume, and then let's say for 500 or 1,000 cases in a big data set. So that can be very difficult. So we wanted to automate all of these steps in an end-to-end workflow. So we came up with this I3CR Vano workflow, which has 4 specific stages. In the first stage, we curate and pre-process the data in a standardized way. In the second stage, we do a multi-class segmentation of the tumor using a CNN. In the third stage, we send this tumor segmentation mask to an expert for review and refinement. And then in the last stage, we convert this final tumor mask into formats that can be easily usable in a clinical setting. The current application, or the initial application of this, was segmentation of pre-op gliomas. And we are extending it right now for other applications as well. So the first step of this framework actually does a scan type classification. So let's say we start with multiple scans which have these varying series descriptions, and we first have a bag of words based classifier which simply looks at the DICOM series description and the number of frames and classifies each scan as non-segmentable and segmentable. And by segmentable, what I mean here is that because our downstream task is the segmentation of gliomas, so we have designed this as a choice that if a scan is one of T1 pre-contrast, post-contrast, T2 or flare, then we will assign it as segmentable. And if it is not one of these 4 sequences, then we will assign it as non-segmentable. So in the stage 1, we have just a binary classification. And in the stage 2, we take only the scans which are segmentable and do the more granular classification into these 4 sequences. So we also determine the orientation, the planar orientation of the scans as axial, sagittal, coronal, and so on. And the scan which has the best resolution or the highest number of frames is automatically selected for downstream processing. And through this process, from every patient session, we have a single scan of each of these 4 sequences. Once we have these 4 scans, then we do a standard set of pre-processing. For example, we register all the scans to a standard anatomical template, and we also do skull stripping to remove any skull area from the scans. And then we have already pre-trained a convolutional neural network for segmentation of gliomas that is deployed within this framework, which we use to produce the segmentation mask of the tumor. Now, as you can see here, that there can be some false positives in the AI-generated mask. And that's why, after this step, we actually send this mask to an expert for review and refinement. So either the expert can look at the mask and they can mark it as all good and they don't make any changes, or if there are false positives like this, they can refine the mask and send it back to us. Now, once we get the mask back from the expert, we compare it with the original segmentation mask using different performance metrics like the dice similarity coefficient for computing the overlap. The next thing that we do is we take this tumor mask and we extract different radiomic features, and we do this using the popular PyRadiomics toolkit. We extract first order statistics like the simple mean, 10 percentile, 90 percentile of pixels, and so on. We calculate shape based features like volume, and we also calculate texture based features. These features are calculated for every tumor subcompartment. So it's calculated for this enhancing, necrotic, edema, these 3 classes, but also the tumor core class, which is the combination of enhancing and necrotic, or the whole tumor class, which is the combination of all these 3 classes, are also of interest. So we calculate the radiomic features for those classes as well. And we do this for each input sequence that are input to the model. And this way we can have at most 1930 radiomic features per patient session from a tumor segmentation mask. And these radiomic features can be very useful for multiple downstream applications. For example, someone can actually use I3CR-Vano on different time points from a patient, and they can simply calculate the tumor volume and longitudinally assess the tumor growth and correlate that, let's say, with clinical decisions. They can also take these radiomic features and build different predictive models, like predicting, let's say, overall survival of the patient, different molecular markers of the tumor, differentiating pseudo-progression versus true progression, and so on. So at a very high level, this is what I3CR-Vano does. You feed it different raw DICOM scans, and there is an end-to-end processing with no manual intervention required. and it outputs a tumor segmentation mask and quantitative information about the tumor. So we evaluated the results for this framework on 384 cases from WashU and 30 cases from MD Anderson. For the scan type classification, we got over 99% accuracy for the different for the 4 classes that we addressed in this framework. And for the segmentation, we also got high mean dissimilarity coefficient for all the tumor classes. You can see here the box plot, which is stratified by the glioma grade. And the different colors represent the enhancing tumor, the tumor core, and the whole tumor classes. So how can someone use this framework in their own data set? So either they can take it off the shelf, they can simply take the framework and start using it. They can, or they can train the models from scratch. So the 2 AI-based components in this framework are the scan type classification and segmentation. So if someone does not want to use our models, they can train their own models and use it within the framework. Or the third way is they can actually take the models that we provide and refine it on their local data. So we have implemented this using Docker for usage in command line. And we have also implemented this using XNAT for graphical user interface. If you are not familiar with XNAT, XNAT is a popular neuroimaging archiving toolkit. And these are the GitHub repositories where we have put all the source code, the pre-trained models, documentation, and some preliminary videos to get started with this framework. So to wrap up, I would mention a couple of things. That first of all, this framework is easily extensible. So in this initial version, we have whatever you see in this red box, that is curation and pre-processing, and we have these 4 scans. We do glioma segmentation, and we do some kind of downstream processing. But let's say we want to do some other kind of brain tumor segmentation, and probably these 4 scans will still be useful for that application. So we can still use this same curation and pre-processing stage, train a different segmentation model for the other type of tumor, and probably have the same kind of downstream processing again. Let's say for organ at risk segmentation, we want to do it on T1 post-contrast scan. Then we can still use this curation, take only the T1 contrast scan, train a model for organ at risk segmentation, and do a downstream processing. The other thing is that we try to make this framework quite modular, and each block is implemented in a each step is implemented as a different block. So let's say instead of the scan type classifier that we are using, someone is interested in classifying diffusion and perfusion scans. They can simply replace the scan type classifier with the diffusion and perfusion scan classifier, as long as the input and output of the steps are similar. For the segmentation model, let's say, as you know, AI is evolving so quickly. So what is the state of the art in 2023 for glioma segmentation will probably not be the state of the art in 2024. So in 2024, when someone wants to use this, they can train the latest model for segmentation and replace the current model with their model. Some of the current directions that I'm working on extending this is we are extending the scan type classifier to include other scan types. We are applying it on other tumor types like segmentation of brain metastasis. And we are also trying it out on neuro-oncology applications, as I mentioned, organ at risk segmentation. We actually have some preliminary results for organ at risk segmentation in a poster that we have later today. So if you are around, please check that out. So finally, this is the publication. This is the published work. So this is the paper that you can check out. I would like to acknowledge all the authors from WashU and MD Anderson for contributing to this work. These are the repositories again, and if you have any questions when you're running this framework, you can reach out to me. And I would like to thank you for your attention, and I can take any questions. Thank you.
All right, we have time for one question.
Thank you, very impressive work. I wanted to ask, what is the false negative rate that you've observed in, or did you have a data set when you're training it to look for possible false negatives?
Yeah, as I mentioned, a couple of AI models. So for the scan type classifier, if I talk about the scan type classifier, then we saw some false negatives in case of FLAIR and T1. So sometimes when it was a FLAIR scan, it was misclassified as a pre-contrast T1, and most of those scans were like very low resolution, low number of frames, and we did not have a scan type classification which was able to handle that. So yeah.
All right, thank you.
Thank you.